Claire, is everything okay?
No, my test on translation is tomorrow and I have no idea what I'm doing.
It's okay. Me and Chris took that last semester. But before you can understand translation, you have to understand the central dogma of biology. Central dogma of biology is the process of RNA being synthesized from DNA and then protein being synthesized from that strand of RNA. Okay, Claire. So after transcription, you're left with a strand of mRNA, which is the form of RNA that is synthesized from the template strand of DNA in transcription. In RNA, the nucleotide thymine is replaced by the nucleotide uracil. The nucleotides on a strand of mRNA are arranged in groups of 3, or codons, and each codon codes for a specific amino acid.
Amino acids, one of which is represented by a blue square, are the building blocks of proteins. There are 20 different amino acids, each with the multiple codons that code for them. Amino acids are carried on tRNA. Each tRNA has 3 nucleotides, which follow base pairing rules with the codons on the strand of mRNA. For this reason, the group of 3 nucleotides is called an anticodon.
A ribosomal unit is comprised of 2 parts and holds the tRNA and the mRNA together. The small ribosomal unit attaches at the start of the mRNA strand, and when it reaches the start codon, which is always the codon AUG, the first tRNA attaches. Then, the large ribosomal unit attaches, completing the initiation complex. The ribosomal unit has 3 sites, the A site, P site, and E site. The A site is where the next amino acyl tRNA waits to add its amino acid to the growing peptide chain. The P site is where the peptide chain currently is, and the E site is where an uncharged tRNA, or a tRNA without an amino acid, exits the ribosomal unit in order to get another amino acid, or in other words, become charged again.
Once initiation is complete, elongation will begin with the second tRNA binding to the A site of the ribosomal complex. Once this happens, the amino acid on the first tRNA will bind to the amino acid in the A site, forming the polypeptide chain. Then, the ribosomal unit will move down the mRNA strand, making the old A site the new P site, and the old P site the new E site. These specific amino acids are the first few in the polypeptide chain that makes up the protein hemoglobin.
The amino acid we just coded for is one of several amino acids that go into the protein molecule hemoglobin. According to MedicineNet.com, hemoglobin is a protein molecule in your red blood cells that carries oxygen from the lungs to the body's tissues and returns carbon dioxide from the tissues back to the lungs. It also plays an important role in determining the shape of your red blood cells as well. Low levels of hemoglobin can be attributed to anemia, which means your blood cells will not be able to carry enough oxygen throughout your body. There are many other ways to obtain this illness, but low levels of hemoglobin is the most common cause. The molecule shown in this picture is the complete molecule of hemoglobin.
So Claire, do you get it now?
Yeah, thanks, that helped a lot.